Ouais, là j'entends le bruit de la salle. Peut-être que je coupe la caméra.
Okay, so good morning, everyone. So thanks a lot for coming to the last seminar of this year. So as usual, people who attend on Zoom, you can report any technical issue in the chat and you can ask your question in the Q&A tab, and we will allow you to interact directly with the room at the end of the seminar. And with no further ado, I give the floor to Cyril to present François today.
Okay, thanks a lot, Jeanne. So I'm really happy today to introduce François Vasseur. François has a very original scientific background. He started his scientific career with a master in crop science and genetics at Rennes. Then he did a PhD at Montpellier at LEPS in a lab in ecophysiology. This is where he started to date Arabidopsis thaliana for a long time, for life. And then he went to Tübingen to a Max Planck Institute to work with Detlef Wegel in a lab in genetics, again in Arabidopsis thaliana for sure, but starting to ask evolutionary questions. And then he went back to Montpellier at CEFE to perform research at the interface of functional ecology and evolutionary biology. And then he has been hired at CNRS for the last 2 years. So, and François, for me, is really the key person to really make the bridge between ecology and evolution. So we are really happy to have him in our group. And so, François, the floor is yours.
So thank you, Cyril, for this very nice introduction. And good morning, everyone. I'm very happy to be here today to present my work in front of the large community of ecologists and evolutionary biologists of Montpellier. So today I'm going to talk about macroecological patterns and microevolutionary or evolutionary processes, and what we can learn from the study of the model species Arabidopsis thaliana. So as an overview, I will first make an introduction about the CSR scheme and how this macroecological pattern can tell us important and informative things about plant strategies and adaptation. Then I will switch to the study of ecological strategies and adaptation in Arabidopsis thaliana. I will then shortly present a way to measure adaptation through the CSR in real time using experimental evolution approaches. And lastly, I will focus on life history traits and the demographic history of Arabidopsis thaliana. So first, an introduction about the CSR scheme and this macroecological pattern that has been described in functional ecology. So a long-term goal in functional ecology, in trait-based ecology, is to explain and categorize plant diversity. So as you can see here, plants are very diverse, ranging from small herbaceous species to large trees. And this variability can be captured by different traits at different organizational scales. And again, a long-term of functional ecology is to categorize plants based on those traits. One of the most prominent models to do that, to categorize plants, is the CSR scheme. the CSR model that has been introduced in the 70s by Philip Grime. And basically the rationale behind this, this model is that Philip Grime considered that there are only two main environmental gradients that led to plant diversification. The first one is an axis of resource availability that I presented here on the x-axis. So basically, on the left, you have environment characterized by high amount of resources, a lot of water, no drought, a lot of nutrients in the soil. And on the right side of the axis, you have environment characterized by a limited amount of resources available for plants, so more stressing conditions for plants. And this limitation of resources is expected to limit growth, to reduce growth, to avoid plant to grow too much. And the other environmental axis of phenotypic differentiation is an axis of disturbance that I presented here on the y-axis. And disturbance is basically any factor that is going to remove biomass, but not reduce growth, but remove biomass after growth. So it's mainly biotic factors, such as herbivores or pathogens. Can be also human factors, such as mowing, for instance. And so based on these two environmental gradients, Philip Grime said that you can identify different types of strategies in plants. The first one is the competitive strategies, so the C strategies, that are located in the bottom left corner of the square presented here, basically in an environment where you have a lot of resources but low disturbance. In the bottom right corner of the square, you can find stress tolerators plants. So basically plants that have been selected in environments that are more stressing in terms of the amount of resources available, but without a lot of disturbance. In the top left corner of the square, we can find ruderal plants, so plants that live in environment characterized by a high amount of disturbance, like agricultural field, like this, but with also a high amount of resources. And in the last corner of the square, Philip Grime posits that there is absolutely no viable strategy that could survive and be maintained in this kind of environment characterized by a low amount of resources available for plants and a lot of disturbance. So of course, a question is how to categorize those plants in this environmental gradient scheme here. And the rationale behind the CSR scheme is that you can use plant traits, to locate any species within this triangle, actually, between the C, S, and R strategies. The idea is that, of course, plants that were selected in, for instance, stressful conditions exhibit different traits than plants that come from more competitive environments. And interestingly, the traits associated with the axis between S strategies and R strategies, so between stress tolerance and ruderalism, is reflected by a set of correlation between leaf traits that has been called the worldwide leaf economic spectrum. And so this set of correlation include different leaf traits, such as leaf mass per area, so the amount of biomass per unit of area of leaves, the nitrogen content in the leaves, the photosynthetic rate of the leaves, and another trait that is not here, that is the lifespan of the leaves. And basically, this set of correlation reflect a trade-off between resource acquisitive strategies on one hand, which are plants that develop leaves that are thin, that are able to photosynthesize very quickly and very rapidly, but because of their thickness, of the fact that they are very thin, they have a short lifespan. So these are typically leaves of crop species, for instance. And at the other end of the spectrum, of the worldwide leaf economic spectrum, we have resource conservative strategies. So basically plants that develop thick leaves with a lot of investment of resources into the structural resistance of the leaves that makes the leaves to live longer for a very long lifespan, but that has a negative effect on the rate of photosynthesis and growth. So again, this worldwide leaf economic spectrum basically decipher plant between resource acquisitive strategies and resource conservative strategies, and is reflected into the SR axis of the CSR scheme. The other axis, basically the one that describe the variation in competitive ability between plants, is a size axis. So I plotted this axis here on the global spectrum of plant form and function, a paper from Sandra Dias in 2016, sorry. And basically, this axis decipher plant between small plants, so short plants with a short stature, small leaves and small seeds, and at the opposite, big plants like large trees and large seeds and large leaves. And this competitive ability, according to grind theory, is mainly driven by variation of size. So, interestingly, it's complicated to measure all the traits related to these different strategies in plants. But there was a recent study from Pierce in 2017 who suggested that actually we can locate any species in the CSR triangle using only 3 leaf traits. Those traits are leaf area, which basically reflect variation along the C-axis, and the covariation of specific leaf area and leaf dry matter content, so the ratio between leaf area and leaf dry mass, or the ratio between leaf dry mass and leaf fresh mass, that basically forms the axis between rudderalism and stress tolerance. And using these 3 traits and interspecific databases, Pierce was able to locate thousands of plant species within the CSR triangle. And for instance, in the red corner here, the competitive corner, you have plants characterized by large leaves that are expected to be more competitive, while in the R corner, you have ruderal plants, mainly annual plants that grow very fast and reproduce very fast. And in the S corner, you have plants that are typical of stressful environments and better to cope with drought, for instance. So overall, the CSR scheme is a very nice model to depict ecological strategies among species, but actually we don't know what about intraspecific variability. This CSR scheme has been developed mainly from interspecific comparisons, and we don't know whether within species where selections occurs, there are such variability in CSR that selection can operate directly on those strategies. So what about selection? Does CSR reflect adaptation to contrasted environments? And also, can we use the CSR, this model, to predict evolutionary response in different environments? So this is typically the kind of questions I'm interested in, because my feeling is that interspecific approaches, comparative approaches led at the interspecific level, are very powerful to identify and describe macroecological patterns. So basically general laws or universal laws of phenotypic variation. However, this kind of approach remains limited to examine the adaptive value of the phenotypic variability we observe between species, and also to test evolutionary processes or mechanisms that could explain this phenotypic variability observed across species. And so to overcome these limitations, I think we need to go to the intraspecific level, so not comparing different species, but different genotypes within species, to unlock those things, and specifically to test the adaptive value of phenotypic variation, and to test as well evolutionary processes. And so to do that, I'm using this very nice model species, Arabidopsis thaliana. So Arabidopsis thaliana has been a model species for 40 years now. It has initially been a model for cellular biology and molecular biology, mainly, but more recently, in the last decade, it also became a very nice model for evolutionary studies and ecology, mainly because it has a large intraspecific variability. So here you have different phenotypes observed between different ecotypes of Arabidopsis thaliana that come from different regions of the world. But more importantly, because we have access to a large data set of genomic information to represent the natural variability of this species. And the main example of that is the 1001 genome data set. So this paper here, where they collected more than 1,000 ecotypes of Arabidopsis, so all the dots you can see on this map, and they fully sequenced them, so you can have now a very nice model to investigate the relationship between genotype and phenotype, and to test also adaptation, evolutionary processes between populations. So I'm going to now switch to the study of ecological strategies and adaptation in this model species, Arabidopsis thaliana. And if we go back to this first pattern, I show you the worldwide leaf economic spectrum among species here. we can measure the same traits, so like leaf mass per area, nitrogen content, or the rate of photosynthesis between ecotypes of Arabidopsis thaliana. So it's what we did in a collection of hundreds of natural ecotypes. And it's what is interesting is that we exactly found the same pattern within species and across species. So the same traits are plotted here, and you find the same set of correlation, suggesting that you have the same trade-off between conservative strategies and resource acquisitive strategies within these species. But the strength of Arabidopsis thaliana is that we get access to the genomic information, and so we can, for instance, identify the genes that are involved in this phenotypic variability. So it's what we did here 10 years ago. Using a QTL approach, we measured those traits and identified the QTL that control the variation of these traits. And we found two main genes with a strong pleiotropic effect, one located at the top of chromosome 1 and one located at the middle of chromosome 5. And those genes have been characterized as flowering time genes, so these are genes that control the development of the plant. And they also correlatively control many traits related to resource economics. So basically, those two genes control the variation in resource use strategies within Arabidopsis thaliana. So how this variation in leaf economics that from what I showed you previously is related to the CSR variation, how does it translate into variation within the CSR scheme between ecotypes of Arabidopsis thaliana? So as I showed you initially, Arabidopsis thaliana is considered as a ruderal species. So it's located on average on the R corner of the triangle, the CSR triangle. But actually, if we measured the traits, and specifically the three leaf traits that I told you before, to categorize plants within this triangle, we found a huge variability between ecotypes, mainly ranging along the SR axis. So basically, you have pure ruderal ecotypes here in blue, but you have also ecotypes that are more oriented toward the S corner of the triangle. So they are more stress tolerators than ruderal ecotypes. And interestingly, again, we have access to the genomic information to test whether this variation is adaptative or not, basically whether it's explained by selection or not. So for that, we use the QST-FST approach. And the idea is to compare the degree of phenotypic differentiation to the degree of genetic differentiation. Because here are the point of origin of the different ecotypes. And of course, these populations are going to differentiate genetically, not only because of adaptive processes, but also because of natural processes, such as genetic drift. So that's why if we measure the degree of genetic differentiation that is measured by this FST metric here, we have an FST that is not equal to 0. So the genetic differentiation is not null between populations, but it's populations slightly differ genetically. At least because of, not only because of adaptive processes, but also because of natural processes such as genetic drift. So it's what we measured here with this distribution of FST. So this is the FST value for all the SNPs, all the polymorphisms along the genome. And the idea of the QST-FST approach is to test whether the degree of phenotypic differentiation measured by this QST is at the very end of the tail of the FST distribution, suggesting that the phenotypic differentiation is higher than what we could expect by natural processes only. And this is exactly what we found here for the R score and the S scores. So basically, those QST for those two traits are very at the end of the tail of the FST distribution, suggesting that there is strong directional selection on those two scores. So what it tells us that, sorry, that in conclusion, we can say that the same physiological trade-offs are observed across and within species. We observed also a wide variation of ecological strategies along the SR axis, less along the C axis, which is presumably explained by the fact that the CSR has been developed at the interspecific level. And of course, Arabidopsis, even if there is variation in competitive ability, this variation will be much, much lower than the variation in competitive ability between species. But it's not the case for the RS axis, sorry. This variation in RS axis strategies and associated with that the variation of the leaf economics is adaptative. There is selection operating on those variations, and that explains the phenotypic differentiation between populations across Europe. And for instance, the S strategy, so the plants that are more able to cope with poor resource availability, which is also associated with resource conservative strategies, are selected in cold regions in the north of Europe. So it's not in the warmest, the driest regions of Europe, like around the Mediterranean Sea, that we can find those S strategies, but it's more in the north of Europe where the conditions are the coldest. So, okay, we have seen that the CSR is able to explain the phenotypic differentiation between populations of Arabidopsis at the large scale, but can we use the CSR to measure evolution in real time, and specifically to test whether rapid adaptation to variation in the environment can be explained by this variation of the CSR strategies? So, to do that, we are currently performing a project, an experimental evolution project, actually, using a large pool of genotypes. So this is the topic of Stefania thesis. She's at the end of her PhD. So I'm going to present you today preliminary first results. But of course, Stefania is still working on that, and we will have a clearer picture of these questions in a few months. So the goal of the project was to test in real time whether we can have adaptation of those strategies in different environments. So basically, we try to simulate the environmental gradient expected by GRIME. So we have two environments that differed in resource availability, no resource availability, or addition of fertilization and watering, and also two other treatments: the absence of herbivores or the presence of herbivores like snails, to simulate the higher disturbance intensity. And the idea is to test whether from a similar population, the same population grown in these different environments, after several generations, we have those populations that move toward different corners of the CSR triangle. So the large pool of genotypes we used for this experiment were composed of actually many crosses, artificial crosses that we did from between different ecotypes of Arabidopsis thaliana. So in this map here, each red dot is natural ecotypes, and the blue lines represent the different crosses we did to generate a large population, what we call a large population of F2 genotypes, that actually represent genotypes that have never been subjected to selection. So we expect to recreate unfit phenotype that have been eliminated by natural selection in natural conditions, but here in our setting, we expect to have phenotypes that are unfit and that we can still measure their traits. And so from this large pool of genotypes, actually 500,000 genotypes, we replicated the same population in different environments. So here it's the field, the experimental field in CEFE, where we simulate those different environments. And we let those populations evolve for several generations. The goal is what we expect to see is that from a trade-off that we can observe between ecotypes, the grandparents of our population, we expect to maybe extend the phenotypic space in this breeding population that has never been subjected to selection. And after several generations of evolution in different environments, we expect to observe ecological specialization. So basically, genotypes that are selected in different environments, and at the end to recreate the phenotypic trade-off we observed in the natural ecotypes. And in parallel to that, we also collect samples every generation in all environments to measure genetic variation, and specifically to test allele frequency variation between environments and between generations. What I can show you is are those pictures where we were able to simulate pretty nicely the environment expected by GRIME. So for instance, here in the environment characterized by a lot of resources and no disturbance, we have many plants that compete with each other. So it should promote accessions with high competitive ability. While for instance, here in the environment characterized by low disturbance, but also low resource availability, we have much less plants. And also interestingly, in this environment that according to GRIME should not allow any viable strategy where there are disturbance and poor resources, actually most of the populations went extinct after 2 or 3 generations, not all of them, but most of them. So it means that we use the right treatment to simulate CSR, the environmental gradient expected by Graham in the CSR theory. As first result, I can show you the evolutionary trajectories of those populations, and specifically the C, S, and R scores in the 3 third generations. What we observed, the colors are not very clear on the board, I'm sorry. But what we see, for instance, what is interesting is that we observe in the 2 conditions characterized by high nutrient and water availability, an increase in the C score for the plants living in those environments. So it suggests again that in those environments you have much more competition, and it selects plants for high competitive ability characterized by a high C score. However, what is surprising is that if you remember what I showed you is that between natural ecotypes, most of the variation is along the SR axis, while here most of the variation we observed in real-time evolution is along the C axis. So we are still working on that to understand exactly how we can explain the differences between what we observed between ecotypes and what we observe in our set of genotypes in control conditions. So now I'm going to switch to life history traits and the demographic history of Arabidopsis thaliana. So there is another trade-off that has been proposed from interspecific comparisons, a trade-off that is expected to be related to demographic strategies, and specifically to the ability to colonize or to compete with other plants. This trade-off is related to 2 traits, actually, seed mass and the number of seeds. So the idea is that, of course, the amount of resources to allocate to reproduction is limited for plants. So plants have to choose between either producing few big seeds or producing many small seeds. And these are expected to have different ecological and evolutionary consequences. So for instance, what is expected is that if plants choose to produce few big seeds, it's because it increases seedling survival and competitive ability. While at the opposite strategy, if plants choose to produce many small seeds, it suggests that it should increase dispersal ability and colonizing ability. So the trade-off between seed mass, individual seed mass, and seed number is expected to reflect a trade-off between colonizing ability and competitive ability. However, this trade-off, first of all, the trade-off between seed mass and seed number has not been very. We lack empirical evidences of this trade-off across species. Here there is almost the only study that show empirical evidence of this trade-off between seed mass and seed number. And secondly, we don't know whether this trade-off really reflect variation in colonizing ability or competitive ability. So as for the CSR, this trade-off between competition and colonization ability is a nice model to depict demographic strategies among species, but we still don't know what about intraspecific variability, what about selection, and whether seed mass versus seed number trade-off really reflect adaptation to different environments. So again, Arabidopsis thaliana is probably a very nice model to study, to investigate these questions. Why? It's because from a genetic investigation, from genetic studies, for instance, the one from me and collaborators in 2017, we can have a nice picture of the demographic history of Arabidopsis thaliana. And for instance, what these authors showed in their study is that during the last ice age, there were climate refugees populations located around the Mediterranean Sea, what we call the relic populations. And after the last glaciation, these relic populations recolonized Europe from the south to the north, as depicted here. But also, very interestingly, those genetic approaches identified a group of genotypes that emerged almost approximately 10,000 years ago in the center of Europe. And these new genotypes rapidly spread throughout Europe and leaves the relict populations only in the very south of Europe, around the Mediterranean Sea, but also some relict populations, or not really relict populations, modern populations, non-relict populations, but with a large part of the genome introgressed with relict old populations. So basically you have this new group of genotypes that spread over all Europe and leaves only oldest populations, oldest lineages at the opposite margins of the distribution range. And importantly, most of the diversity of Arabidopsis we can find today come from this group of modern genotypes that emerge in Central Europe. 90% of Arabidopsis in Europe are from this group of genotypes. But we don't know how these genotypes and why these genotypes were so successful in colonizing Europe after 10,000 years ago. And so, to investigate this question, again, Arabidopsis is a very nice model to test differences in competitive and colonizing abilities. So to investigate this question, we performed an experiment in greenhouse. Actually, this experiment was performed by Cristina Bastias, a former postdoc, and Aurélien Estarag, a PhD at the end of his PhD. He's going to finish his PhD at the beginning of next year. So they performed an experiment 2 years ago, where they used 70 natural accessions of Arabidopsis, categorized in 5 geographical groups. The relict accessions in orange here, yes, in orange here, that are only in the south and mainly in Spain. Non-relict south accessions, so accessions that come from this modern group that emerged in Central Europe, but that coexist at the same places than relict populations. The group of central accessions in green here, and 2 groups in the north of Europe, the high north populations, which are close to the relict populations, genetically at least, and another north group of populations, more intermediate between the high north and the central one, that we should expect should also be intermediate in terms of phenotype and genotype between those 2 extreme groups. And they measured plant traits related to dispersalability, like the plant height, the number of seeds produced, and the size of the seeds. And they also measured the response of plants to intraspecific competition, as well to drought. The first thing they showed is that there is the same trade-off, the trade-off between seed production, the number of seeds, estimated here by the number of fruits times the size of the fruit, as there is a trade-off between seed mass and the number of seeds. So this expected trade-off from interspecific comparison is really well observed within species. And more importantly, the different geographical groups are located at different positions within this trade-off. So you can, for instance, easily see here that the blue dot here, so the high north non-relict populations, are at the one end of the spectrum, together with the south relicts that are here, while the center non-relict central populations are mainly, so more spread, at the other end of the spectrum. So if we look more in details at this variation in terms of fecundity, plant height, and seed mass, we observed a very nice correlation between the geography and these traits. So again, center non-relict populations, so those again that colonize Europe, are more fecund, they produce more seeds, but smaller seeds, and they have higher reproductive stems. They are taller. So compared to the extreme populations that come from the both edges of the distribution range of the species, which, for instance, the relicts and the high north non-relicts are exhibit less seeds, but bigger seeds, and at the same time, they are also smaller. So basically, and also we measured the response to competition and the response to water stress. And interestingly, again, as expected, the populations from the center of Europe are the more impacted by competition, while those characterized again by big seeds, but few big seeds, are less impacted by competition, for instance, than the Central European populations. Oh, sorry. So again, it suggests that first, there is a trade-off between seed size and seed number in Arabidopsis thaliana, and it well reflects the demographic history of Arabidopsis. At least it explains it. It explains why those Central European populations were so successful to colonize Europe. It's presumably because they have traits related to dispersal ability, associated to better response to competition, and to a lesser extent, a better response to water stress. While the extreme populations, those that come from the margins of the distribution range, produce less seeds but bigger seeds, and they are better for competition. We use again the genomic information of Arabidopsis thaliana to go a bit deeper and to measure the FST between populations. And for instance, what we observe is that there is a huge FST for seed mass. So here is the black line here. And more interestingly, so here is the FST between all the populations, south non-relict, center non-relict, mid-north non-relict, and high north non-relict, compared to the relict populations only. And so what we can see is that there is an increase in genetic differentiation for between relict and Central European populations, while this differentiation decreases when we go upper in the north. So what it suggests is that those populations that come from the north are closer to the relict population, and specifically for the genes related to seed mass, because here is the FST measured on the genes that control seed mass. And again, we went a bit deeper on this analysis by measuring the enrichment in outlier alleles in these different populations. So this comes from a previous studies when they measured the amount of outlier alleles in the different populations of Arabidopsis thaliana. And what they observed in previous studies is that there is an enrichment, a strong enrichment in outlier alleles in relict populations, which is expected because those relict populations are very different from the rest of Arabidopsis thaliana populations. And so here we measured whether there is an enrichment more important in the genes related to different processes. So here is the relict, and for instance, there is a strong enrichment in outlier alleles for the genes, or the alleles related to seed mass, for the alleles that have a positive effect on seed mass, exactly the same that we observe for the high north relict populations, and the opposite was observed for the more central populations of Arabidopsis thaliana. So basically, it suggests that when those populations spread from the center to the margins of the distribution range, they reach environment where this very successful dispersal ability phenotype become less and less favorable, and they introgress some alleles of the relict populations from the south or from the north to better cope with environmental variation, and notably with stress in those extreme environments at the margins of the distribution range. So as conclusion, we can say that there is also an intraspecific trade-off between seed size and seed number, as observed across species. But more importantly, this trade-off shapes the genetic and phenotypic differentiation of Arabidopsis thaliana across Europe. And the colonizing success of central genotypes can be explained by their high fecundity and dispersal ability at the expense of competitive ability and resistance to stress. And also what is important and quite interesting is that we observe the phenotypic and genetic convergence at the range margins. So both at the south of Europe and at the north of Europe, we have similar phenotypes that are selected although the environmental conditions are very different in those two regions. So in general conclusion, we can say that trait relationships are similar across species and within species, at least those that I presented today. But more importantly, the macroecological patterns that have been described and intensively described at the interspecific level can inform about the microevolutionary processes and mechanisms operating within species. More important also, moreover, ecological strategies related to resource use are selected along a latitudinal gradient, while life history traits that shape the demography history of Arabidopsis thaliana show more a center to margin gradient. So what's next? So if you remember this plot I presented earlier, with this nice breeding population we use for our experimental evolution experiment, Stefania analyzed the phenotype of natural accessions, so the red dot in this map. And interestingly, she identified a group of natural accessions that we can consider as phenotypic outliers. So they are really at the edges of the phenotypic space when we compared all the accessions, the natural accessions of Arabidopsis thaliana. So interestingly, these outlier, the phenotypic outlier, this functional outlier, come from two regions, one in the north and one in the south, but not at the very far north of Europe. There are only 2 or 3 genotypes here, and not at the very south of Europe. So basically, they are kind of intermediate between Central European populations and relict or relict-like populations in the north. So it could suggest that these phenotypic outlier that we observe in these regions could be located in contact zone between relict and non-relict populations. And so this phenotypic outlier maybe could result from recent hybridization between relict and non-relict populations. So this is a very recent result actually from Stefania PhD. And we are currently trying to test that in more details. Notably, Katarina Utami just started her PhD 2 weeks ago to investigate these questions in Arabidopsis thaliana. So that's it. I want to thank all of you, of course, but I want also to thank all my collaborators for who participated to this work. So as I mentioned during the talk, Stefania, Christina, and Aurélien, but also 2 former supervisors, Denisville, my PhD supervisor, and Cyril, my postdoc supervisor. We collaborate a lot on that, and so it's really a collaborative work. And also, I want to thank all the EcoPart team for the very nice meeting and discussion we have all the time in this group. I want also to take the opportunity of this talk today to say that I'm looking for postdocs. So if you are interested, short advertisement, do not hesitate to contact me if you're looking for a postdoc. Thank you.
Questions?
Do you have any information about the shape of leaves? I asked this question because 20 years ago, we measured the shape of different ecotypes, and we have seen that the ability to compete with oneself was determined by the shape of the leaves. Okay. So I would be interested to know whether you measured this trait, the shape of the leaves, with all the accession you have here.
So your leaf morphology or leaf shape between ecotypes. Some colleagues did that, and so I think there are plenty of data in the literature to maybe investigate whether leaf shape could at least explain part of our responses. So that's an interesting point. I never thought about that, but we can have a look at that. That's definitely interesting.
Yeah, so you presented this graph where you had the strong correlation in traits for the natural accessions, but when you cross them, you expect to modify the pattern of variation. And do you really observe that? Because I don't think you've shown data about that, and I was interested in seeing if there's actually indeed a lot of variation that is created by this outbreeding in a pure, mostly selfing species, whether you see that or not.
So no, it's what we expected, but so I didn't want to include those results, but because again these are very preliminary. But Stefania is working on that currently, and so she measured the phenotypic space based on different traits between natural accessions and those F2 populations, and actually they are very, very similar. So initially we had two hypotheses to explain what we could observe in breeding populations like these F2 populations. Either we show that So basically, there is no genetic or phenotypic constraints. And so if you combine alleles by crossings, you amplify the phenotypic space. It was our new hypothesis, we could say. Or there are strong constraints on the phenotype, on those correlations, genetic or phenotypic, biophysical, I don't know what kind of constraints. But basically, it would suggest that it's impossible to escape from these trade-offs. And it's more or less what we observe. So basically, we observe the same trade-off in the F2 population than in the natural ecotypes, which suggests again that it's very difficult, by at least from genetic mixing of alleles, to break those trade-offs.
And also maybe related to that, so I think it was described, I mean, there was some strong cases of strong outbreeding depression described in Arabidopsis when you cross accessions that have been isolated for long. And do you see that, or do you lose those lines anyway because they don't survive and you don't have them?
It's a bit early to answer, actually. What I can say from something that I didn't present today, we measured hybrid variability. So from these crosses, so here it's F2 populations, but to produce F2 populations, first we had to create F1 hybrids that we phenotyped. And we observed variation, I mean, it's very contrasting depending on the hybrids. Some hybrids show strong heterosis, so they perform better than their parents. And some hybrids show kind of depression, inbreeding depression, sorry, and they perform clearly less well than their parents. So there is no clear patterns that emerge from the analysis of these hybrids.
No, the case I knew of were at the F2 level.
At the F2 level.
Yeah.
So it's a bit early to answer this question. Stefania is still working on that. We just have now measurements of fitness. And so we can try to address this question, but I have no answer right now. Sorry.
Questions?
So you said that the Arabidopsis is a very nice model. I think I agree now. Do you think it's the best model to challenge macroecological questions and at least how to bridge interspecific observation and intraspecific test? Do you think that Arabidopsis is enough, or what we could do more?
I think Arabidopsis is a very nice model because we have access to a wide range of information about phenotypic differences between ecotypes as well as genotypic differences between ecotypes. But there are other species for which we have the same kind of information. Lolium perenne, for instance, is one of them, but also crops. But crops is a bit different because crops have been strongly selected for specific traits. So it reduces a lot the range of phenotypic variability. So yeah, for me, Arabidopsis remains one of the best model to test this kind of question. But there are other species for which it could be also performed and for which we could also try to understand whether what we find in Arabidopsis is also true for other species. And also, I think one way to also to make the bridge between interspecific comparisons and intraspecific studies like this, it would be to include relative species to this kind of study. So basically, we know some close relatives of Arabidopsis, like Arabidopsis lyrata, Arabidopsis allieri, which are very close, and we know when they diverge from Arabidopsis, for how long. They have also different, they express different traits, and specifically important evolutionary transitions, such as the variation between annual type and perennial type. So looking at intraspecific variability between close relatives of Arabidopsis, but in any species, for me would be also very interesting to understand how to switch from intraspecific level to interspecific large-scale comparisons. Yes, please.
I seem to remember a study that was done here at ISEM, Nicolas Gatier's group with Jonathan Romiguet. I think that one of their findings was something also related to number versus size of offspring that you do, and this was a predictor of genetic diversity across species in animals. I think so. So I was wondering, did you look at any effect of this trade-off at nucleotide diversity within this in these different, or do you find differences in nucleotide diversity in these different accessions? And or can you do that within accessions? I guess you can, right? Nucleotide diversity, you can do that within an individual, I guess, well, through heterozygosity or something. But
I think we have all the tools to do it because, again, we have access to the full genomic information. We have plenty of trait measures across thousands of ecotypes. So we can do that, but we haven't done that yet. So that's definitely something interesting to do.
So I don't know, someone can correct me, perhaps Nicolas.
Actually, I don't.
Do you remember that, or
we'll have a look at it.
That was the finding that they had, or something? Yeah.
Okay, but we could check.
The idea is that species with a small number of big propagules, they are more resilient to bottlenecks, to say it briefly. So to find this within Thaliana, you would need to have some kind of history of isolation that you have. So
we could try that.
Maybe.
Interesting.
Maybe if some population, but it's something I didn't plan to ask the question, but it's something I did not really understand in your explanation of the history of the populations, because maybe it's because it's a selfing species and it's different from outbreeding species. But usually adaptive alleles, they can spread in space completely independently of the neutral genetic background. But here it seems that you have this genetic background, the central background that spread to get together with the adapted genotypes without admixing too much with the relic populations. And maybe you have reproductive barriers as suggested by Ophélie or something like that?
Yes. So there are several elements to answer this question. So first of all, yes, I think part of the answer is because of the sulfur reproduction of Arabidopsis that limit a lot gene fluxes between populations. On average, the outcrossing rate in Arabidopsis is estimated between 5 and 10%. So there are still some level of admixture, but very, very low. The other thing about reproductive barriers is that we know that these central populations have also different developmental rate and flowering time than the peripheral populations. So they flow at different times. So maybe it also participates to limit the gene fluxes between populations, because basically there is a low chance to cross between them because they don't flow at the same time. So it's another part of the answer.
So if you have reproductive isolation, then you can expect that the populations that have been resilient to bottlenecks are those with few big propagules when compared to many small propagules.
Yeah, that will be particularly interesting because those populations are at the margins of the distribution range. So it's that's very interesting. Yeah.
Just 2 points, but it's worth to verify.
So last question by Nicolas.
So thank you. Just a question about the effect of. It seems that you talk about the effect of genes on different traits related to fitness. So maybe you can show the slide again, because I'm not sure I understood how these were measured, and also, Yeah, exactly this
Okay,
one.
so, yeah,
Maybe you can
faster.
explain a bit, and so my question is about if these alleles, they are the same in the south and in the north, or not, and it was very fast, and so I couldn't really understand quite well what you were saying.
Yeah, sorry if I went too fast. I agree, it was a bit fast. So I'm going to try to take a bit more time to explain that. So from previous studies, they measured the amount of what they called outlier alleles in the different genotypes of Arabidopsis thaliana. And what they observed is that in relict populations, because they diverge from the rest of Arabidopsis thaliana population a long time ago, they are more different, and so they are more outlier alleles. Outlier because they are from relict populations. So more or less, we could say not outlier alleles, but more or less relict alleles.
Yeah, it's just the demography.
Yeah, and actually, so here it's the enrichment, but we can also measure the amount of outlier or relict alleles in the relict populations and in the other populations. Of course, it's not much higher in relict populations than in the rest of Arabidopsis genotypes. So basically, along the genome, there is a certain amount of outlier alleles in the different genotypes of Arabidopsis thaliana. This amount is higher in relict populations. Okay, this is the context. So then here, what we did is that we measured the amount of outlier alleles in 1,000 genes, SNPs, taken randomly along the genome. So this is the amount we should expect for the background, the genomic background, the amount of outlier alleles we should expect for the genomic background. And then we also measured the amount of outlier alleles in 1,000 SNPs associated to fecundity, seed mass, plant height, or the response to water stress, or the response to competition. And so then we measure the difference between those two things. So whether it's what we call the enrichment, is whether the genes, the SNPs that are involved in the variation of those traits show more abundant outlier alleles than the genomic background.
But are they also equally spread along the genome, or could it be located in some regions that are over-differentiated between populations?
We don't know. We haven't measured that. I can try to have a look at that.
Because depending on the location of the genes, you don't expect the cell level of FST.
So it's not FST.
No, but I mean.
Okay. Here we just wanted to measure whether the SNPs that are associated with specific traits are enriched or depleted in relict alleles versus the rest of
It's just I'm not sure about your normal
so
to do that.
How they are, where they are located along the genome, we haven't really looked at that. So maybe we could.
It's just that your normal is just across the genome. But I mean, oftentimes for people, I don't do, but some people do FST scans, and we know that if you just take this as your normal, you can get outliers quite easily. So I was just wondering about this.
Okay, so yes, if you have some papers or whatever, I would be interested to have a look and to have a better test of what we do. But what we can still say is that okay, maybe there's some bias in the way we performed our analysis, but still we have a very consistent pattern with our expectation. So we have an enrichment in the north for the alleles that have a positive effect of seed mass, as expected, while we have a depletion in the central populations. And we have the opposite pattern, for instance, for the alleles related to plant height. So there's a depletion of alleles having a positive effect on plant height on the far north, and no effect on the central population. So as far as I understood, if there was a bias, it would be more or less random. There is no reason to think that an increase in outlier, because we didn't take into account everything, would result in such a nice geographic pattern, I guess.
So you wouldn't expect contact to be large in the south, because it seems that it's different in the north than in the south.
You mean from that? No, actually, the phenotype in the north and in the south are quite similar. The relict and the high north phenotypes are quite similar.
But you don't see conjoined in the south on the graph.
Yeah, it's here. Sorry. Here it's the relict. But the scale is not the same for relict. So you have, of course, enrichment that goes up to 15%. Here you have south non-relict. And so the pattern is not exactly the same, I agree. But again, these south non-relict populations are genetically very, very close from the center non-relict, much, much closer to the center non-relict than to the south relict population.
Any other question? Right, thanks a lot, François.
Thank you. Je peux pas trop vite. Ça a été. Ouais.
Ça fait un bon panorama de tout.
Ok, bon,
Ouais,
merci.
c'est bon, vas-y.
Bien.
Fallait pas en mettre un.
Trop long.